Hi everyone, my name is Elisa Elizondo.
My name is Jessica Yeager.
My name is Dylan Shoemaker, and we're going to talk to you about a really important global virus called cassava mosaic virus that's causing devastating problems to both central and western parts of Africa.
African cassava mosaic virus is found anywhere cassava is grown in Africa, mainly the sub-Saharan region, but it's also been found in India. This virus is widespread and destructive in the humid lowlands of Africa. It was originally named Krauskrankheit, which is German for crinkling illness. The name is indicative of the symptoms observed. Presence of this disease is identified by mosaic molting, decreasing or no production of tubers, distorted leaflets, reduced leaf and plant size. Cassava susceptibility varies by cultivar, but all cultivars can be infected.
African cassava mosaic virus was first described in northeast Tanzania in 1894. It was discovered in the late 1930s that the disease was transmissible, and the idea of it being a virus was proposed. It wasn't until 40 years later that its viral status was proven and the mode of transmission was discovered. The disease can be transmitted from infected stem cuttings, white flies, or by grafting infected budwood onto healthy plants. White flies are the most common form of transmission. The adult whitefly needs 3 hours to acquire the virus in an 8-hour latent period, and then it takes only 10 minutes to feed on a new plant and infect it. The virus stays inside the whitefly for 7 to 9 days and even stays in the insect during its molting process. The youngest plant leaves are the most susceptible and infected part of the plant. African cassava mosaic virus is a Gemini virus, meaning it has small twin particles. Each particle has one molecule of single-stranded DNA, and the genome consists of 2 circular molecules. The virus particles cause damage by accumulating in the nuclei of phloem cells in the outer cells of leaf tissue, causing major problems for sugar, nutrient, and water movement in the leaves of the plant.
The most common forms of management for this virus involve rouging, the removal of diseased plants, intercropping and planting of resistant varieties and virus-free cuttings. Unfortunately, controlled planting and chemical control are not practical for the farmers of Africa to control the whitefly vector. Whiteflies typically prefer to feed along the field margins, and in order to counteract that, resistant varieties are planted around the border of the fields. Farmers can also plant alternative hosts to attract the pest.
There are multiple strategies being studied to prevent and control cassava mosaic virus. Some methods currently being researched include developing resistant varieties through traditional plant breeding and genetic engineering. Molecular analysis shows that the gene CMD2 confers monogenic resistance to the virus. This single gene explains over 60% of the resistance to CMV. Using backcross breeding, the resistance gene has been backcrossed from landrace varieties into cassava accessions. Landrace varieties of TME9 and TME117 identified near the virus's origin of southwest and central Nigeria were used in traditional breeding projects. The broad specificity of the CMD2 gene suggests that varieties conferring resistance to cassava mosaic virus may also show resistance to other pathogens affecting this important food crop. Besides traditional breeding programs, another research project for control of cassava mosaic virus is through the use of genetic engineering. Using a hairpin loop from the coat protein in cassava brown streak disease, resistance has been established to both cassava mosaic virus and cassava brown streak virus. The inserted hairpin was derived from small RNAs and inserted into cassava. This infection forms an established mode of resistance to viral invasion. Once either pathogen tries to replicate in its host, the small interfering RNAs will target the mRNA, attacking and disassembling the pathogen. This mode of resistance prevents replication of both cassava mosaic virus and cassava brown streak disease, and serves as a form of virus-induced gene silencing. In a study of co-inoculation of both cassava mosaic virus and cassava brown streak disease, no viral symptoms were observed for over 7 months. The generous financial donations through organizations such as the Bill and Melinda Gates Foundation are helping bring the scientific knowledge to the farmers in these developing nations. These funding efforts to support continuous scientific investigation through both the public sector with universities such as Cornell and the private sector through companies such as Corteva Agriscience are helping lead to a future where this pathogen is a problem of our past.